Welcome everyone. Today I'll be solving question number 36 of the 2016 GATE Biotechnology paper. The question is that a 1.2 kb DNA fragment was cloned into the BAM H1 and EcoR1 site located on a 2.8 kb cloning vector. The BAM H1 and EcoR1 sites are adjacent to each other on the vector backbone. The vector contains a ZO1 site located 300 base pairs upstream of the BAM H1 site. An internal ZO1 site is present in the gene sequence as shown in the figure. The resultant recombinant plasmid is digested with EcoR1 and ZO1 and analyzed through 1% agarose gel electrophoresis. Assuming complete digestion with EcoR1 and ZO1, the DNA fragments visible on the agarose gel will correspond to. There are four options given to you. Option A is 2800, 700 and 500. Option B is 2800, 700 and 800. Option C is 2500, 700 and 800. Option D is 2500, 1200 and 300. So let's see out of these which option is correct. So this is the gene of interest and this is the vector. So the recombinant DNA will look something like this. This will be the BAM H1 site. This will be the EcoR1 site. This will be the ZO1 site, which is internal, and the vector ZO1 site. The distance between the vector ZO1 site and the BAM H1 site, as we have told you, is 300 base pairs. The distance between the BAM H1 site and the ZO1 site inside the insert is 500 base pairs. The distance between the ZO1 site and the EcoR1 site is 700 base pairs. What is remaining of the recombinant vector is 2.5 kb. So if you digest this recombinant DNA with EcoR1 and ZO1, you will get three cuts. One will be here at the EcoR1 side and two at the ZO side, one in the vector itself and one in the gene of interest. So you end up with three fragments. One will be the fragment between ZO and EcoR1, which will be 700 base pairs. One will be from ZO1 to ZO1, which will be 300 plus 500, which is equal to 800 base pairs. And what will be remaining is 2.5 kb. That is 2.5 kb or 2500 base pairs. So the answer in this case will be option number C, 2500, 700 and 800. That's all for this question. I hope that was easy for you to understand. Thank you so much for listening.